Let me get rid of that for a second. All right. For the work that led to his two laws of inheritance, Mendel chose pea plant characters that turn out to have a relatively simple genetic basis. Each character is determined by one gene, for which there are only two alleles, and one's completely dominant over the other, and it's completely recessive. But these conditions are not met by all heritable characters, and the relationship between genotype and phenotype is rarely so simple. Mendel himself realized he could not explain more complicated patterns he observed in crosses involving other pea characters or other plant species. The inheritance of characters determined by a single gene deviates from simple Mendelian patterns when alleles are not completely dominant or recessive, when a particular gene has more than two alleles, or when a single gene produces multiple phenotypes. Alleles can show different degrees of dominance and recessiveness in relation to each other. In Mendel's classic pea crosses, the F1 offspring always looked like one of the two parental varieties because one allele in a pair showed complete dominance over the other. In such situations, the phenotypes of the heterozygote and the dominant homozygote are indistinguishable. Some genes, however, neither alleles completely dominant, and the F1 hybrids have a phenotype somewhere in between those of the two parental varieties. This phenomenon is called incomplete dominance and is seen when red snapdragons are crossed with white snapdragons. All of the F1 hybrids have pink flowers. So in this case, a heterozygote phenotype is a blend between the two parental homozygous phenotypes. And so we see what was called an intermediate or in-between color in the offspring. Now, when you crossbreed those F1s, those colors will sort back out in the F2 generation. So here we have a red, homozygous red, crossed with a homozygous white, and we get pink. When we cross these two pinks, you can see that in the F2 generation, we see red flowers, pink flowers, and of course, white again. The same thing can be true with hair texture. So the curly versus straight, not completely dominant over one another. So if you have an allele for both, you're going to have wavy hair. Another variation on dominance relationships between alleles is called codominance. In this variation, the two alleles both affect the phenotype in separate, distinguishable ways. So in this case, neither allele is dominant or recessive, so both parental phenotypes will be seen in all the F1 offspring. One example for humans is sickle cell anemia, or the sickle cell trait. So normal cells versus all sickle. If you have if you're heterozygous, you will have both types of blood cells floating around in your bloodstream. Here's a chicken example with a white and black chicken, and then we have what's called a speckled black and white, and the same thing for roan cows and horses. So in this case, the dominant or recessive will always be either or, so red or white. Codominant will show both. This is red and white, and this is incomplete will blend to make a third new color. So that's a blend. So again, incomplete dominance, blending, codominance, both show together. So let's just see. So if we look at this column here, here are the genotypes. What type of pattern would be exhibited by this example? Well, that would be complete dominance. They're either blue or yellow. The next set of weird little faces, we have blue, yellow, and yellow. That's going to be codominance. And then the last one, we have blue, yellow, and blue make green. That's going to be incomplete dominance. Multiple alleles refer to genes that have more than two alleles. If there are four more possible phenotypes for a trait, then more than two alleles have to exist in that population. However, individuals are only going to have two of those alleles because they have two parents. Human blood groups show multiple alleles and codominance. The ABO blood groups in humans are determined by three alleles for a single gene. We refer to that as IA, IB, and then lowercase I. A person's phenotype or blood group can be one of four types: A, B, AB, or O. These letters refer to two carbohydrates, A and B, that may be found on the surface of the red blood cell. A person's red blood cell may have carbohydrate A, giving you type A blood, carbohydrate B, type B, both AB, or neither, which is type O. So here we have, this is pretty cool. This is complete dominance, codominance, and O is the lack of these antigens or carbohydrates, and so they have none of those. And the A's and the B will always be dominant to O. So far, we've treated Mendelian inheritance as though each gene affects only one phenotypic character. Most genes, however, have multiple phenotypic effects, a property called pleiotropy. In humans, for example, pleiotropic alleles are responsible for the multiple symptoms associated with certain hereditary diseases such as cystic fibrosis and sickle cell disease. Now, I do want to show you this real fast. So here's how the sickle trait is inherited. Both parents have the sickle cell trait. So again, it's a recessive allele, and in order to have all sickle cells creating sickle cell disease, you have to inherit both recessives. But here's the complication. So this is what we're talking about. Because of having sickle cell disease, you're going to have increased pressure is going to cause the cells to sickle some, low oxygen levels, dehydration, high altitudes. Here is sickle cell with athletes. So the cells are misshapen and they can't carry oxygen as well, and so that's going to cause other complications in the body just to the shape of the cells. In epistasis, a gene at one location alters the phenotypic expression of a gene at a second location. An example will help clarify this concept. In mice and many other mammals, black coat color is dominant to brown. For a mouse to have brown fur, its genotype must be little b. But there's more to the story. A second gene determines whether or not the pigment will be deposited into the hair. The dominant allele, symbolized by capital C, results in the deposition of either black or brown pigment, depending on the genotype at the first location. But if the mouse is homozygous recessive for the deposition gene, little c, then the coat is white and it expresses as albino, regardless of the genotype at the black-brown location. In this case, the gene for pigment deposition, big C or little c, is said to be epistatic to the gene that codes for black or brown pigment. Polygenic traits result from the interactions of two or more genes. One of those examples is eye color. There are several genes that contribute to eye color, not just the simple brown's dominant to blue. There's evidence that human skin pigment is controlled by at least 3 separate inherited genes. Let's consider 3 genes with a dark skin allele, where dark skin is all capital, contributing 1 unit of darkness to the phenotype, and is incompletely dominant to the recessive allele version of that. So a person who has more dominant alleles is going to be very dark, while a person who has more recessive alleles will be very light, and someone who's heterozygous for both of those, all of those genes, would have an intermediate shade as their skin tone. Another departure from simple Mendelian genetics arises when the phenotype for a character depends on the environment as well as the genotype. A single tree locked into its inherited genotype has leaves that vary in size and shape and greenness depending on exposure to wind and sun. For humans, nutrition influences height, exercise alters build, sun tanning darkens the skin, and experience improves performance on tests. Even identical twins who are genetic equals accumulate phenotypic differences as a result of their unique experiences. Another example is sea turtle sex determination. This is pretty cool. So the female will lay the eggs in the sand, and depending where they're located in that nest, the further down they are where it's cooler, these will be males, and then you have the mid-range will be the females, and then the warmer the temperature will also be the males. So in this case, the environment determines the sex of the sea turtles. Another example are hydrangea flowers. they're directly affected by the pH of the soil. So you can see in this instance, these are